Welcome everyone to today's lecture.
Attila, we cannot hear you for some reason.
Now.
Yeah,
I can hear you.
Okay, so once again, I wish to welcome you all to today's seminary talk here at the University of Debrecen, Department of Evolutionary, Zoology and Human Biology. Today's guest is Professor Nick, used by Dr. Lendvai Adam. Adam, the floor is yours.
Okay, thank you very much. I'm really honored to introduce Professor Nick Lane from the Department of Genetics, Evolution and Environment of University College London. To be honest, it feels like a bit when a bunch of teenagers meet their favorite pop star, so I don't think that he needs much of an introduction, but I still say a few words about him. So Professor Lane is a biochemist who studies how energy flow has shaped evolutionary history. Most of his current work is on the origin of life, but his research spans from the evolution of sexes and the female germline to mitonuclear co-adaptation and redox signaling to the evolution of eukaryotic cells. He has published many research papers in leading journals, including Nature, Science, and Cell, but he's probably best known as the author of 4 amazing popular science books that has received various prestigious awards and have been translated into close to 30 languages, including Hungarian, although as far as I know, only one of them has been published, sadly, into Hungarian. I really do hope that at least the vital question will get translated into Hungarian as well, so that the wider audience can read it in Hungary as well. Not just his books, but he also received a number of awards, including the Biochemical Society Award and the Royal Society Michael Faraday Prize. Not as if his research wasn't exciting enough, but he looked extra excitement and thrills in rock climbing for many years. I don't know if you still do that.
Not much anymore, I'm afraid.
Unfortunately. And if you guys are lucky, you might also stumble into a London pub and see him playing the fiddle. Probably not. Actually,
I'd still do that.
Okay, so actually that's the name of the band, Probably Not, which is hilarious. Today, he's going to talk about energy and matter at the origin of life. Please welcome Professor Lane.
Well, thank you very much. That's a really kind introduction. Let me share my screen. Can you see that full screen?
Yes, we
let
can.
me also have a pointer. Okay. Well, thank you very much. It's really a pleasure. So I was just saying to Adam before, the origin of life is a field which really should be grounded in biology, but for a long time has been grounded in chemistry. And in many respects, it is a question in chemistry. But the chemists have a different set of goals and ambitions, and really what they're interested in is making the building blocks of life, and what biologists are interested in are quite a different set of questions. So I'm going to give you an overview of how I would see the field, some of our work and some of the context for that. So we immediately run into the problem, if we're thinking about the origin of life, as to this question, what is life? And Schrödinger had a famous book on this entitled What is Life in 1944. And my own take on this really is he had 2 main themes in that book. One was to do with genes and information, and he was the first person to talk in fairly modern terms about a code script, for example. This was during the Second World War. It was at a time when encryption was very much in the air. And so his terms are quite modern in far as genes and information are concerned. He also famously talked about entropy and talked about life feeding on negative entropy. And here his terms are quite antiquated to my mind. He talks about life continually sucking orderliness from its environment. And it's very hard to imagine what he really had in mind or what that means in biological terms there. But he did have a footnote in the second edition to his book. And it was, if I'd been catering for physicists alone, I should have let the discussion turn on free energy instead, which I think probably makes much more sense to most biologists. So just to make sure we're all on the same page, what is free energy? Well, it's the energy available to power work of any description, from muscle contraction to enzyme function and so on. And in our own case, it comes from respiration, from burning food in oxygen, in effect. And respiration is generating entropy in the universe as a whole, heat, but we are conserving free energy in the form of ATP, adenosine triphosphate. There's nothing particularly special about the bonds in ATP. It's really the ratio of ATP to ADP, which is skewed 10 orders of magnitude from equilibrium, so that there's far more ATP relative to ADP. And it's that disequilibrium which is powering work in cells. And we are turning over, a tremendous amount of energy. We're turning over about our own body weight in ATP every day. Is there then a universal form of energy conservation across all living things? ATP is often called the universal energy currency, but it is formed almost universally, or pretty much universally, by the ATP synthase and by proton gradients across membranes, electrically charged membranes. So an electrical membrane potential, proton gradients across the membrane driving ATP synthesis. Amazingly, these proton gradients are as universal as the genetic code itself. I say that deliberately because there are a few trivial exceptions to the universal genetic code, and of course there are a few trivial exceptions to this statement about membrane bioenergetics being universal. There are a few cells that are basically fermentative, but very few, and they tend to be at the tips of the tree of life. But this machinery of membrane bioenergetics, I've mentioned the ATP synthase, and membranes themselves are enormously complex and would appear on the face of it to be perhaps almost irreducibly complex. So how could they possibly be prebiotic? It's a question which has meant that most people have backed away because it's not obvious where the answer might lie. The assumption has been that there must have been simpler forms of energy conservation at the origin of life. And these are what most of the field talks about most of the time. So we start with molecules like cyanide that have a lot of energy pent up in the triple bond between carbon and nitrogen. Related compounds like cyanacetylene, UV radiation as a form of energy generation, lightning, famously going back to the Mille-Urey experiment, wet-dry cycles and so on. This is the kind of environment that most of the field, certainly the chemists, are thinking about these days, what are known as terrestrial geothermal systems, things like Yellowstone National Park, where we see these kind of systems, where you have an interface between air, between the water, where you have a possibility of drying, where you have UV radiation and so on. And it certainly offers scope for a lot of interesting chemistry. But that chemistry has been done over decades, and at the end of it, there is very little resemblance between the prebiotic chemistry that's been done and biochemistry as any biologist would know it. To the point that I would call this Frankenstein chemistry, the unnatural origin of life. There's almost no overlap between the kind of chemistry that has worked in these experiments over decades, I won't go through this list here, and biochemistry as we know it. So I think there's a serious problem here of joining up. How do we join up the origin of life to life itself? Now, maybe that's a spurious question. It was addressed theoretically, at least, by Christian de Duve, and I like the way he was thinking about this question. Of course, this is not a proof of anything, but he asked, how did proto-metabolism come to be replaced by metabolism? The obvious answer to this question is that the appearance of catalysts, whether ribozymes, protein enzymes, or both, was responsible for the transition. He then says we have to ask how catalysts with appropriate properties came to appear. And the answer is a really interesting one. He says the only scientifically plausible explanation, so if we assume that God didn't do it, is that catalysts arose through selection. And he then finishes by saying enzymes are only going to be selected if they fit into proto-metabolism. So he's assuming more or less a seamless transition from geochemistry into biochemistry, and that the first biological catalysts effectively enhanced flux that was happening anyway in some geochemical environment. I think as a biologist, that makes a lot of sense. And the first question we have to ask, though, is well, is there is it possible to go from some kind of geochemical flux proposed by de Duve into biochemistry as we know it? And that means, first of all, we have to ask, well, what is the most ancient forms of biochemistry? And this probably won't surprise very many people. This is a tree of life from Bill Martin, a rather conceptual tree of life from about 20 years ago now, a little over 20 years ago. And there's only a couple of things I really want you to see from this. This was very radical when he put it out. And it's more or less, I won't say mainstream now, but certainly it's more believable now than it was 20 years ago. So two things to notice. One of them is that the eukaryotes, basically all morphologically complex life, arose through endosymbiosis. This is the chloroplasts, an endosymbiosis with cyanobacteria giving rise to algae and plants. And down here is a rather singular endosymbiosis between an archaeal host cell, or population of archaea, and bacterial endosymbionts. And now, when he put this forward, I doubt that many people really would have thought of this as a singular endosymbiosis. The discovery of the Loki Archaeota and the Asgard superphylum and so on over the last 5 to 10 years or so does suggest that really something like this did indeed happen. What this means is that the eukaryotes have no relevance to the origin of life. They are a derived domain, and the last universal common ancestor down at the bottom here is the common ancestor of bacteria and archaea, of prokaryotes. And what Bill has down at the bottom is two separate emergences of these groups, these domains of life, from a common ancestor that lived in a hydrothermal vent. And that's another radical proposition. Why on earth would he even say such a thing? Well, it's because the bacteria and the archaea are shockingly different to each other in many respects. So plainly, they share a common ancestor. Because bacteria and archaea share the universal genetic code, ribosomes, the basic structure of ribosomes, many of the enzymes involved in transcription and translation and so on, implying DNA and RNA, and of course the genetic code. Quite a lot of intermediary metabolism, but not necessarily always the genes, but some of the genes in parts of the Krebs cycle, for example, and membrane bioenergetics. So having an electrically charged membrane driving ATP synthesis, but also other things like CO2 fixation, which I'll come to. So why then two separate emergences? Because the things that are different are quite really surprising. The cell membrane, it is equivalent in terms of biophysical properties, in terms of its conductance, for example, its thickness, its general electrical properties. But in terms of the chemistry, they're very different. Along with the enzymes required to synthesize the lipids, the cell wall as well. So the barrier between the inside of the cell and the outside world is basically not homologous in bacteria and archaea, which begs the question as to what kind of membrane or wall did the common ancestor have? Pathways like glycolysis that most people would assume were ancient. The chemistry is conserved, but the genes that encode that chemistry are not actually conserved in bacteria and archaea. Even DNA replication. So unlike transcription and translation, DNA replication, most of the genes and the enzymes involved are not homologous in bacteria and archaea, suggesting that it arose independently on two separate occasions or something else happening. Whatever the answer is, the question is an interesting and difficult one. Even respiratory chains, these are more difficult because they can be passed around by lateral gene transfer, but it looks as if there may have been two separate origins of pumping in bacteria and archaea. So at the very least, this begs the question, what kind of a cell was this last universal common ancestor? And it brings me to a paradox. And the paradox is that membrane bioenergetics are universal, but the membranes themselves are not. There's a lovely quote from Niels Bohr about paradoxes. He says, "How wonderful that we've met with a paradox. Now we have some hope of making progress." And I like paradoxes partly for this reason. And now in this particular case, the paradox could be resolved. There are probably several ways of resolving it, but it could be resolved if life originated in an environment where there were geological proton gradients. And of course, there is such a place. It was discovered 20 years ago by Deb Kelly, who was captain of the Alvin submersible in the year 2000. And she discovered this lost city hydrothermal vent field about 15 miles away, 15 kilometers away, let's say, from the mid-Atlantic ridge. And it's very different to the kind of events that most people are familiar with, the black smoke events, where you see the smoke belching out of the top of the chimneys. And she's holding here in her hand a lump of a black smoke event. And you can see there's a chimney, and this is where the smoke is belching out. And here she's got a lump of this lost city. And it's much more like a sponge, a mineralized sponge. It appears to be a very porous kind of rock. So it's formed in a very different way to the black smoker type vents. Black smokers are formed by the interaction between water percolating down and interacting directly with magma. And so they're basically volcanic systems. They're formed at the mid-ocean ridges and so on. These alkaline hydrothermal vents, like Lost City, are formed off the axis. It's water percolating down into the submarine crust, and water can percolate down 5 or 6 kilometers, I'm told, into the crust. And there it can react with minerals that are rich in ferrous iron and magnesium, ultramafic minerals like olivine. And they react with that. The iron is oxidized to ferric iron, and it's an exothermic process. So it's producing warm water, warm meaning about 70 to 90 degrees rather than the black smoke events, which are often 300 or 400 degrees. And they're strongly alkaline, about pH 11, and generally bubbling in hydrogen gas. Now, these kind of vents really should be forming on any wet rocky planet where there is plenty of olivine. And in the last 10 years or so, it's transpired that that seems to be the case. So this is Enceladus here, and these plumes which jetting out into space, about 200 kilometers into space. The Cassini flyby showed that they are alkaline in pH, about pH 10, and they're rich in hydrogen gas and various organics, including methane. It seems as if processes of serpentinization may be going on underneath the liquid ocean, underneath this frozen crust on Enceladus. Similar things happening on Europa. And occasionally, traces of methane have been detected on Mars, and there's always some hopefulness that might suggest life on Mars. Is more likely, if anything, if those traces of methane really are robust and true, it's more likely that trace amounts of water are reacting with olivine to generate methane by serpentinization. But hopefully we'll find out for sure one way or another in the next few years. These vents were proposed to exist, were hypothesized about 10 years before they were discovered by Mike Russell. And he has been interested since the late 80s in the origin of life, and the work that I will talk about is grounded in his ideas. We don't agree about all of these things, but certainly he was an inspiration to the way that I think about these questions. So this throughout the 90s, he was not well known. These were marginal ideas, but the discovery of Lost City meant that suddenly his view became mainstream because he predicted that this kind of hydrothermal system would exist. And this was in 2009. There was a feature article in Nature entitled Naissance Man. They've dressed him up as a Renaissance man, as Erasmus, and Naissance playing on the origin of life, of course. And here there's a bit of Lost City behind him, and this is a hydrothermal reactor that he's built at the Jet Propulsion Laboratory in Pasadena. The key points, though, this is the structure of Lost City, and Mike had been arguing that these would have been much more rich in the Hadean period 4 billion years ago in minerals like iron sulfides, iron nickel sulfides. The oceans were much richer in dissolved metals because there was no oxygen around, and there would have been high concentrations of hydrogen, as there are in modern systems. Far more CO2 in the oceans, giving them a mildly acidic pH, maybe between 5 and 6. And that would give proton gradients across catalytic walls containing iron sulfide minerals. So he saw these as electrochemical flow reactors at the bottom of the ocean, potential incubators for the origin of life. And I find these really very appealing ideas. I find them appealing precisely because they link to bioenergetics. They link to this idea of membrane bioenergetics specifically, and the questions that I have spent my whole well, adult life really thinking about and being interested in how on earth did these systems, these complex systems, arise in the first place. So this is a cartoon of a bacterial cell or something along those lines, and it's just keeping it to the absolute minimum that we've got some kind of a membrane pump, which is pumping protons out. We have a membrane, which is a standard lipid bilayer membrane, and pumping protons out means it's relatively alkaline inside and relatively acidic outside. And if we convert the electrical potential into pH units, that works out at a pH difference of about 3 to 4 pH units between the inside and the outside of a modern autotrophic cell. And here we have a pore in a hydrothermal vent. This is very conceptualized, but we have acidic ocean waters percolating in, as I say, a pH of 5 or 6. And we have alkaline hydrothermal fluids, which would be about pH 11. We must expect mixing within the vent, but it's at least possible to imagine that some places in these vents may have had, across a thick inorganic barrier, thick relative to a cell membrane, still quite flimsy, the pH gradients of 3 or 4 pH units across a barrier. So the magnitude is similar and the polarity is similar, acidic outside, alkaline inside. So the question is, can we imagine, can we test any way in which we could go from this topological system of inorganic system to an organic cell? Is there any way that we can cross that gulf? So the first thing we need to think about is, why on earth do cells use proton gradients in this way? It's a very strange idea. And it took a long time for people to come around to accepting it. This was known as the Ox-Phos Wars, as in oxidative phosphorylation, really from the early 60s until the late 70s when Mitchell won the Nobel Prize. But there were still questions raging until the late 80s about mechanisms of pumping and so on. Even now, there are still questions about how all this works. So there's a lovely quote from Leslie Orgel, who was a pioneer of origin of life works, who said, "Not since Darwin has biology come up with an idea as counterintuitive as those of the great physicists, Einstein, Heisenberg, or Schrödinger." I should say that, that Mitchell himself, this is 1947, with Jennifer Moyle. So Mitchell was an ingenious thinker, but he was apparently cack-handed in the lab. And he won the Nobel Prize by himself in 1978, which I'm increasingly thinking was unfair to Jennifer Moyle, because she did most of the experiments which established certainly Mitchell's own research agenda. And I think that really she should have been acknowledged far more than she in fact has been. But returning to Mitchell himself, there's a quote from a conference that he went to in 1957. So this was before he put forward the chemiosmotic theory. And this conference was on the origin of life, the invitation to Moscow from, I'm forgetting his name now, Oparin, from Oparin. And he says, "I cannot consider the organism without its environment." So when I say organism here, think bacteria, don't think about multicellular organisms. So I can't consider the organism without its environment. From a formal point of view, the two may be regarded as equivalent phases between which dynamic contact is maintained by the membranes that separate and link them. So I'm reading this carefully because it's an extraordinary statement. He's saying that the difference between the inside of a cell and the outside world, that these are equivalent phases, and they're separated and linked by a membrane which is 5 nanometers thick between the inside and the outside world. And he's talking about separating and linking them. So it's very, very carefully phrased. Now, it may seem a peculiar way of seeing the world, but if you're thinking about the origin of life, before there is such a thing really as the inside of a cell, a living cell, or the outside world, these are phases, these are equivalent phases. And it does seem to me to be very important to think about the difference between these two phases and what is crossing membranes. So I find this a really helpful way of thinking about the origin of life. Now, Mitchell at the beginning was not thinking about respiration. He really came from a background of working on bacteria and bacterial physiology. And this question about, well, how do bacteria effectively keep the outside out? How is it that these two equivalent phases are in fact quite different to each other, the inside and the outside? And he recognized, it seems obvious to us today, that membrane proteins would have to have the same finesse as enzymes. They would have to recognize their substrate with as much sensitivity as an enzyme, or would have to actively pump it out very often against a concentration gradient, so it's going to cost energy. And of course, if you allow it to flow in again, in the case of protons, then in principle, at least, it's possible to conserve some of that energy which is released as protons flow down their own concentration gradient. And I'm saying here, this Mitchell took this mental leap to go from bacterial physiology to respiration in humans, and realized that the membrane and proton gradients and so on are necessary in respiration. But also in bacteria and archaea, they are involved in CO2 fixation. And this is something I'm going to say more about shortly. So why CO2 fixation? Most ancient cells, if we believe phylogenetics, probably grew from hydrogen and CO2. It's very difficult to get at the deepest branches of the tree of life by phylogenetics, but there are various comparative biochemistry reasons as well to think that potentially the deepest branches in the methanogens, which are archaea, and the bacteria, which would be acetogens, are both living from this reaction between hydrogen and CO2. This is the only pathway of CO2 fixation which is found in both bacteria and archaea. And there are some quite deep differences even in the way that this pathway works between these two groups, implying that it really was a very early divergence rather than a later lateral gene transfer. So both groups are growing from hydrogen and CO2 in this way. In the case of methanogens, to release methane as a waste product. In the case of acetogens, to release acetate, acetic acid, as a waste product. And overall, not the first few steps, but the last few steps especially, it's exothermic. So the delta G naught prime is minus 131 kilojoules per mole. We don't need to worry about that if you're not comfortable with these chemical terms. It's basically exothermic. It should happen spontaneously. And if we consider what we know about the core of metabolism, beginning with hydrogen and CO2, the kind of metabolism that we see in methanogens and acetogens and many other anaerobic bacteria that appear to be deep branching. I just really want to bring your attention to a couple of things here. Hydrogen is marked in a kind of brown color here, and hydrogen, everywhere you look, there's hydrogen. Nowadays, it would be coming through NADH or NADPH or from ferredoxin or something, but it's basically hydrogen which is being transferred here. And then CO2 in green, and again, hydrogen and CO2 are really at the roots of all this intermediary metabolism. We're starting at this side with activated CO2, and within a few steps of the acetyl-CoA pathway, we get to acetyl-CoA, not to acetyl-CoA in a prebiotic environments, but to some form of activated acetate, potentially just acetic acid bound to a mineral surface or something. And then we're into the reverse Krebs cycle, or at least parts of it anyway, an incomplete reverse Krebs cycle, pyruvic acid, oxaloacetate, alpha-ketoglutarate, making amino acids directly in a single step very often, aspartate, for example, glutamate through to glutamine, and sugars, glyceraldehyde, ribose. The blue arrows here are experiments that have been done under prebiotic conditions following these pathways. So quite a lot of this has been done, albeit not under a consistent set of conditions. These are different groups working with different conditions, but quite a lot of this chemistry really does work from hydrogen and CO2 under prebiotic conditions. The other thing to draw attention to here, in the red boxes, these are the precursors for nucleotide synthesis. We normally would use ATP now rather than acetyl phosphate, but this is an energy currency, just 2-carbon energy currency that might be able to do that job instead, and I'll return to this later on. So we don't have to go very many steps into metabolism to get to the precursors for nucleotide synthesis, and I'll return to that right at the end of the talk. Why would all this happen? Excuse me. Well, it happens in part because thermodynamically it's favored. So this is some theoretical thermodynamics from more than 10 years ago now, from Tom McCollum and Jan Amond, showing that under these alkaline hydrothermal conditions, this is the delta G, The more negative it is, the more favored it is. So this is total cell biomass at 50 degrees centigrade. And all this is really showing is that under these alkaline hydrothermal conditions, if you start with CO2 and hydrogen, it is favored to make total cell biomass. If you were to shake them together in a test tube, thermodynamically, what we should get is cells. Especially we should get amino acids and fatty acids. Nucleotides, notice they're up here, they're above 0. This means it's endergonic. You need to put some energy in to make nucleotides. But if you can conserve some of the energy from down here and put it in up there, then overall the total cell biomass is exothermic overall. So we know in principle that this should happen. In practice, we know that it doesn't happen. And the reason for that is that there is a barrier. I will call this an energetic barrier. It's somewhere between being a thermodynamic and a kinetic barrier to the reaction between CO2 and hydrogen. And as I say, it's overall to go through to methane, it's strongly exergonic. To get to the redox potential of most of organic chemistry in cells is just slightly exergonic. But to get there, there's this quite steep uphill gradient to get through. This is formaldehyde at the top here. So it's steeply uphill to get to formaldehyde. And once we're through that, it's downhill all the way to methane. Now, what methanogens are doing is using a proton gradient across a membrane to lower this kinetic barrier to the reaction between hydrogen and CO2, making it far more likely that we would indeed get these kind of conditions. So the question then is, are there geological environments that can drive this? And we've already seen that that's possible. Here's what methanogens do with these proton gradients. This is a membrane protein. It's an iron sulfur protein called the energy converting hydrogenase. It sits in the membrane. Here's the ATP synthase, which is a far more complex protein. The ECH takes the electrons from hydrogen and passes them on to ferredoxin. And ferredoxin is then capable of reducing CO2. So this is overall not a favored reaction. There's about a 200 millivolt shortfall in reduction potential for this reaction to happen. And it draws on the proton motive force. It draws on the proton gradient across the membrane to power this reaction to get over this energetic barrier so that hydrogen is able to pass its electrons on to ferredoxin. And so the question is, can we imagine an abiotic way of doing something like this? And yes, we can just through physical chemistry, really to begin with. Here's the problem that we face, that at pH 7 or any other pH, hydrogen is not reducing enough to reduce CO2 through to formaldehyde. Technically, this is becoming more negative as we go up, so it's an uphill reaction. That's all you really need to take home from this. Hydrogen is unable to force its electrons onto CO2. This is this barrier, the first couple of steps. But that's at pH 7. If we think about the conditions in vents, we actually have hydrogen at pH 11, which makes it more reducing. And we have CO2 at pH 6, which makes it more easy to pass the electrons onto. The reasons for this are quite intuitive. If hydrogen at pH 11 passes its electrons onto something else, then what's left behind are protons, and protons being formed in an alkaline environment will react immediately with the hydroxide ions to form water. So it's a neutralization reaction, and that's thermodynamically strongly favored. So the more alkaline it is, the more likely it is that hydrogen will pass its electrons onto something else. It becomes more reducing. Hence this gap. This is just the Nernst equation is about 60 millivolts per pH unit. And for CO2, it picks up an electron, it picks up a negative charge. If it can balance that charge by picking up a proton, then it can pick up another electron and so on. So it's much easier for CO2 to pick up electrons to be reduced in an acidic environment where protons are available. And again, this is the Nernst equation just applied to the standard reduction potentials. So in principle, so long as these phases are kept separate, in other words, so long as there are two phases separated by some kind of a barrier like a membrane, it should be possible to transfer electrons from hydrogen to CO2. And it's possible to imagine then potentially protons crossing this barrier and driving this reduction of CO2 by hydrogen to form organic molecules. And if that were happening, we can imagine again an endpoint where we have some kind of a protocell wedged inside this, where we kept keeping the topology that I've talked about, where instead of having protons crossing the barrier, we now have them interacting with some kind of iron sulfur clusters, perhaps linked with short non-coded peptides or amino acids or something sitting in a fatty acid bilayer membrane as a first step. So We're keeping the topology. This is now beginning to look much more like a testable hypothesis, and we can begin to do some experiments. I've been trying to do experiments on these kind of questions for 10 years or more now, and we started out with very simple systems, trying to build relatively large reactors, thinking that we're simulating hydrothermal systems, so they should be big. In fact, they turned out to be almost uncontrollable. We found it really difficult to repeat our own experiments. Here we've got precipitating little vent systems inside the reactor. And so we ended up trying to do microfluidics instead, which allow us to control the actual specific conditions much more closely. And we've had some positive results, enough to make me think it's possible to do this. I'm not going to go through these in any detail because we've basically failed to repeat most of our own experiments. This was the most exciting one. This shows formaldehyde up to tens, if not hundreds of micromolar over a couple of hours. But we were unable to repeat our own experiments, so I've never published that. If you're interested afterwards, you can ask me a question and I'll explain exactly what went wrong and why we struggled. But it's been surprisingly difficult. But a former student of mine, about a year ago, working with Ruben Hudson, this was Victor Soho as my former student, has demonstrated finally, using a microfluidic chip, that hydrogen in an alkaline phase will pass presumably electrons, but this is not fully established yet, onto the barrier, and that CO2 in the acidic phase on the other side will pick up those electrons and protons to balance the charges and can be reduced to formate. Excuse me. So this happens over a very short period of time, just a few seconds. And Ruben and Victor had shown using NMR that indeed the electrons are coming from hydrogen, the CO2 is really being reduced to the CO2 that's added, where they were using 13 CO2, so isotopically labeled CO2, and the protons using deuterated water really were coming from the water on the acidic side. There are various other possible interpretations about what was crossing this barrier, and I'll come back to that in a moment as to whether it's protons or electrons crossing the barrier. At the moment, we're not absolutely sure. But what this showed beyond any kind of doubt is that if we have two phases with a barrier separating them, then it will facilitate the reduction of CO2 to form organic molecules. It didn't work if there wasn't any hydrogen. It didn't work if there wasn't a barrier. It didn't work if there wasn't a pH gradient. It didn't work if it wasn't mildly pressurized. It's only 1.5 bars or so. So it corresponds very closely to the conditions that we were thinking. I have to say, this is only the first step. It's only a few seconds, but formate is not very far into intermediary metabolism, and I think we can and should be doing much better than this. But at least it's a proof of principle. One thing we've been trying to do is look at the possibility of whether it's protons or electrons crossing this barrier. And this is a microfluidic chip without a barrier at all, with laminar flow, and you're able to maintain a pH gradient even with laminar flow. And as soon as we have barriers, very often these barriers can be quite diffuse, with plenty of water inside the barrier itself. And one thing that struck us quite early on is that the barrier tends to become acidic. Even here, wherever we see water inclusions within the barrier, they tend to be acidic. And we looked into that in more detail. So this is using pH sensitive dyes. This is the blue is the alkaline, the yellowy green is the acidic. So it's about pH 6 here, about pH 11 here. This is the beginning of the barrier and all kinds of other bits of barrier, but you can see right up to the very edge, it's acidic. And by doing some stopped flow experiments, I won't go into the details here, we were able to show that we can have very steep gradients, as much as 3 or 4 pH units across single nanocrystals of iron sulfide, McKinowite, about 25 nanometers in length. And we were able to show by the stop flow experiments that protons will cross this barrier about 2 million times faster than hydroxide ions going the other way. So it does look as if these barriers will tend to facilitate the transfer of protons. And that means at least this arrangement that I've proposed already is in principle at least feasible. But there's a lot of questions there. Let me just come back to this. We have here, we have proton gradients across barriers. So we've got acid on one side crossing that barrier. So acidic potentially here. We've got strong alkaline conditions. And we're not anticipating that these are very complex protocells. We're thinking about Fischer-Tropsch type synthesis of fatty acids. And so these vent conditions are often considered far too harsh for protocells, fatty acid vesicles to exist in. So the pH range we must think about is pH 5 through to 12, temperature range of 50 to 100 degrees centigrade, salinity, probably modern ocean type salinity, that's the best guess, even for the Hadean period, 4 billion years ago. And then high concentrations of divalent cations like magnesium and calcium, which can play havoc with fatty acid bilayer membranes and effectively turn into soap. So we needed to look to see, is it even possible? So there was an interesting experiment from Tom McCollum more than 20 years ago now, where they started with formic acid, so formates. This is basically where Victor's experiment finished. And this was at about 150 degrees centigrade under stronger hydrothermal conditions. The idea was to break down the formate back into hydrogen and CO2 and react them together under these conditions. And what they found was long chain hydrocarbons. This is 10 carbons in the chain, 11, 12, and so on. These are hydrocarbons. These are alkanoic acids in the black here, and alkanols. So basically fatty alcohols and fatty acids and long chain hydrocarbons. So it's perfectly possible to get in this kind of experiment chain lengths of about 10 to 15 carbons in a chain, and quite complex mixtures of these under those conditions. Interestingly, it only happened in a steel reactor. It didn't happen in a glass reactor. Why steel? It's presumably the steel itself is exerting some catalytic effect, and it may be the carbon ion, the carbide bonds in steel that are doing that. And we may also see those kind of bonds in hydrothermal systems as well. So the fact that it only works in steel doesn't necessarily mean that it's not important for the origin of life. So we took some of those mixtures and we looked to see if they would form bilayer vesicles. And most of the literature in the field has been on single fatty acids vesicles, decanoic acid, for example, so 10 carbons in the chain. And this is optical density. So at higher pH, you tend to form micelles, and micelles are tiny things that don't scatter light very much. And here at more acidic pH, everything gets protonated, and you end up with with effectively fats, uncharged fats, which form globules, and that scatters light a lot. So vesicles are forming in this intermediate region here, and you can see with a single fatty acid that they're stable around about pH 7, but there's very little range on that. As soon as we have more complex mixtures of fatty acids, 6 different fatty acids here, we're beginning to extend that range up to about pH 9 or so, from pH 7 up to pH 9. And if we have mixtures of fatty acids and fatty alcohols in a 5-to-1 ratio, we're up to around pH 10. And here, this is a 1-to-1 mixture of fatty acids and fatty alcohols. And rather than having these rather gracious curves, we see this mess. We were quite shocked to see that. We didn't really know what it meant, but if we take it at face value, it implies that we should be getting vesicles everywhere. And that was in fact the case when we use, this is cryo-electron microscopy, and these are bilayer membrane bound vesicles. And here we see with confocal microscopy, again, we get plenty of vesicles. This is pH 12, 70 degrees centigrade, and modern ocean salinity, including these divalent cations. So it's perfectly possible to get stable vesicles forming. In fact, it seems to be easier to form them under these conditions. So the idea that we can only form them at pH 7 and nowhere else is not true. We can indeed form stable vesicles under fairly harsh hydrothermal conditions. The other thing which amazed me and we were delighted about, we were wondering if we had mixtures of simple amino acids, cysteine, for example, with the iron sulfur minerals, would we be able to make these minerals smaller, more biological in context? Would we be able to chelate iron sulfur minerals using cysteine as a ligand? And rather surprisingly, it turned out that not only does that work, but at alkaline pH, at pH 9, where the thiol in cysteine deprotonates, we get 4Fe, 4S clusters. This is UV Vis spectroscopy, and there's a classic shoulder on UV Vis spectroscopy. We were able to demonstrate that it depends on the concentration of cysteine, it depends on the pH. Mossbauer spectroscopy shows again that we get different mixtures. So we're getting 2Fe, 2S, 4Fe, 4S clusters, and then various other types of cluster. The higher the cysteine concentration, this is 10 millimolar cysteine concentration, we get less complex mixtures. We get mostly 2Fe, 2S clusters and mononuclear clusters, whereas here we're getting a lot more 4Fe, 4S clusters. And these are redox active. They're not particularly reducing. We need to do a lot more work to see, to try and separate these mixtures and work out what are the actual properties of different types of cluster. But the surprising and rather beautiful thing is that you're able to form biological 4Fe, 4S clusters, the same thing that we see in ECH ferredoxin, using just a single monomer cysteine as a ligand. And I mentioned earlier on nucleotide synthesis. So quite a lot of this core intermediary metabolism has been done under these kind of hydrothermal type conditions, but nucleotide synthesis never has been. This is a PhD student, Stuart Harrison, who's been looking at the formation of uracil. So this is the nucleobase rather than the nucleotide. And here's just focusing on these last few steps from dihydroorotate through to uracil, using a range of different metal ions from the first step. So going from dihydroorotate to orotate, and for example, getting a relatively high yield with molybdenum here. And then across at the far side, this is uracil, and interestingly, we get quite a lot of uracil in the presence of copper sulfate. No other metal ions seem to work at all, but copper, and it appears to be a free radical mediated reaction, and various other things are capable of doing it as well, other than metal ions. But copper is able to do it. When we optimize the conditions for this red box steps going from dihydroorotate to uracil, we tried different pH, we tried different temperatures, we tried different pressures, we tried different salinities and so on. We tried a range of these things. We were quite agnostic as to which conditions might work. And we found that we could optimize the yield about 6-fold of uracil. This is the standard conditions at the beginning compared to the conditions that we'd optimized to. And this was high pressure, modern ocean salinity, alkaline conditions about pH 8 to 9, and high pressure and warm temperatures, 90 degrees centigrade. So it's interesting that it corresponds to these alkaline hydrothermal conditions as well. And this was a one-pot synthesis of uracil, orotate and uracil, starting over here with carbamoyl phosphate and aspartate, which are the biological precursors. And this is switching pH between, because some of these reactions are facilitated at lower pH, some at higher pH. And when we switched pH, which is the kind of thing we can do in a hydrothermal vent just through percolation, then we get there. It's not a very high yield, but it is a yield of uracil under those conditions. Here's another rather beautiful set of experiments that I'd just like to mention briefly. Why is ATP the universal energy currency? We tried using acetyl phosphate and a bunch of other different potential phosphorylating agents to phosphorylate ADP in water. And this is what we see. So these are multiple different phosphorylating agents, and acetyl phosphate works pretty well. We get 15 to 20% yield at pH 5.5 to 6, lower yield at pH 7, and nothing really at pH 9. This is 30 degrees C, this is 50 degrees C, and again, we're getting a good yield at 50 degrees C, but it's being hydrolyzed then much faster, probably the acetyl phosphate itself being hydrolyzed. Again, CP is carbamoyl phosphate, and nothing else works apart from cyclic trimetaphosphate, which is here. So it seems to work. This is in the presence of ferric iron as a catalyst. It didn't work. We tried about 10 different metal ions, and it didn't work very well with any of the other metal ions. Ferric iron works much better than anything else. Here's the really beautiful thing. This is phosphorylating ADP to ATP in water. This was the control at 0 hours, and there's some contamination of ATP in the ADP from the manufacturers. but we get quite a big increase in ATP over the period. When we do the same thing with ITP, GTP, CTP, UTP, we find that we don't get any increase over the control, so the contamination in the manufacturer's product. So we're able to phosphorylate ADP to ATP, but none of the other bases. So there's something favored about adenosine, which is presumably one of the reasons why it became adopted as a universal energy currency. It can be done in water. And this is pyrophosphate, and again, it doesn't work with pyrophosphate. So we were able to come up from various experiments that I won't show you with a potential mechanism, and it's quite interesting. Ferric iron it's small, it's got a very high charge density. It's got half the electrons in the d orbital, or half the orbitals are full. Whatever the reasons are, we're not absolutely sure, but this does not work on surfaces or under many other conditions, but it's capable of bringing acetyl phosphate close enough by interacting with the diphosphate and probably with this amino group on adenosine. This is the key to why adenosine works and the others don't. So this is really a condensation reaction in water, and it's often described as impossible. And it's worth again saying that if we have pH gradients, then it becomes not just possible, but almost inevitable, because it's as simple as a titration. And here's an example, just a very simple one to give you a feeling. If we've got this as a barrier here, if we have electrons being transferred onto it, we have a leaving group of a hydroxide ion in acidic conditions will react with a proton to form water. In other words, this dehydration reaction of forming water in water is perfectly possible if the leaving group is a hydroxide ion and it's under acidic conditions. Then we have simply a titration reaction, and it becomes not a difficult thing to do at all. So the question really, again, is we're messing around with pH. Under what conditions will pH differences across membranes, across barriers, promote these reactions to happen? Right, I think I've got just a few minutes left, and I'd like to finish with the origin of information. This is some really theoretical thinking about how these things might go on and where the genetic code itself comes from. So this is a protocell in some kind of a barrier. We're reducing CO2 through a prototype ECH to give a C2 intermediate, something along the lines of acetyl-CoA, but an activated acetate, going into a reverse Krebs cycle. And from there, we're making fatty acids, an energy currency equivalent to acetyl phosphate, sugars, various amino acids. And so this is a branching proto-metabolism, the kind of thing I showed you earlier on. The question is, can it get better at doing these things before there is a genetic code? And the answer is, well, if there are positive feedbacks, it's possible in principle at least. So to make nucleotides requires sugars, it requires amino acids, and it requires an energy currency. But once we've made a nucleotide, there can be positive feedbacks, for example, on CO2 fixation. In this case, the folates, for example, in acetogens, or molybdopterins in methanogens are involved in CO2 fixation. They're derived from GTP, so they are effectively naked nucleotide cofactors capable of catalyzing specific reactions. Here we've imagined these nucleotides feeding back on all of these other different pathways. And here we're imagining them feeding back on nucleotide synthesis itself, so an autocatalytic loop. So this is a mathematical model just to give us a sense of the architecture of these systems. And what we find very briefly is that if this is a strong catalytic feedback on here, then we have a faster rate of protocell growth. All of these components are made and they divide in two once we've doubled the surface area. If it's a weak catalysis, of course, we don't really see any difference at all. So in principle, if we have positive feedbacks, a nucleotide feedback on CO2 fixation will promote protocell growth. Interestingly, if we have feedbacks on all of these other pathways, it generally doesn't work very much and will often collapse growth, because if we are, let's say, feeding back on amino acid synthesis, then too much flux comes this way and not enough flux goes other ways, and effectively it crashes protocell growth. So long as we're keeping the cell as the selective unit and thinking about growth as the important aspect, then nucleotides, unless they're balancing across these pathways, will not promote protocell growth. It's an interesting point when we think about how nucleotides work in life today. They're very often ATP, for example, or NADH. They're doing quite general jobs that would work across all of these pathways. And the final one then is this autocatalysis of nucleotide synthesis. What we were hoping was that nucleotides would accumulate under some set of conditions that way and would be then more likely to polymerize. What we actually find is that this is autocatalysis on the x-axis. This is catalysis of CO2 fixation. So it's doing both this and that. And if it's doing them both well, maximum rates of each, then we get maximum rates of protocell growth and division. But the way in which to accumulate nucleotides is when it does autocatalysis, but doesn't really drive CO2 fixation very well, and that means that we have slower rates of protocell growth. So it's interesting. What it tells us is that there aren't very many conditions if we're dealing with cells as a selective unit where nucleotides are going to accumulate to very high levels. So just for the last 2 slides, I think now, there are various patterns in the genetic code that have been known about for decades, but few people have thought about them from the point of view of autotrophic protocells, the kind of thing I've just been talking about. So let's assume that we're beginning to polymerize nucleotides inside protocells. What are we going to get? This is a map of metabolism, and this is a pattern that's been known about since at least the 1980s. The first letter of the codon, in this case, it's a G, it's shaded gray where it's a G at the first letter of the codon, corresponds to the precursor of the amino acid that it codes for. It's basically a single step reductive amination from a carboxylic acid in each of these cases, from oxaloacetate to aspartate, from pyruvate to alanine, from a methylene to glycine, and from oxoglutarate to glutamate. In yellow, it's an additional step, and it usually involves ATP, and this is A at the second position of the codon. In green, in blue, I'll say first, we have cytosine at the first step, cytosine at the first step of the position of the codon, and in green, it's uracil. And all I really want you to take away from this is that there is a relationship between the first base in the codon and the distance from CO2 fixation in this basic autotrophic map of metabolism. It's a fairly significant relationship. Exactly what it means is harder to say, but it certainly is not random. Here we see if we have a G at the first position of the codon, so in gray here, or an A at the first position of the codon, the ones in yellow here, then at the second position, we see a strong relationship with hydrophobicity. This is the hydrophobicity of the amino acid and the hydrophobicity of the base, and we're seeing a strong correlation at that second position. This is based on quite a large number, 43 different ranking scales of hydrophobicity. They're all a bit different to each other, and we see this strong relation. If there's a pyrimidine at that second position, we don't see that kind of relationship at all. Again, all I really want you to take home from this is this suggests that there are direct biophysical interactions in the code, and that it's not random. And then finally, in the third position of the code, again, we're seeing this is an inverted codon table, and I'm just going to highlight one example here, that if we have a G at the first position, an A at the second position, and a purine at the third position, then we get a glutamate. Whereas if we have a G and an A and then a pyrimidine at the third position, we get an aspartate. So this is usually considered to be more or less redundant at the third position, but it matters whether it's a purine or a pyrimidine at that third position. And again, all I want you to notice from here is it is not random. It depends on the size of the amino acid, and again, it implies that there are specific associations between amino acids and the anticodons specifically that are coding for them. We are trying to write up a paper on this at the moment in terms of what are these relationships, what can we understand from them. It's quite abstruse, but the real take-home message from this is that these are not random relationships. There are biophysical interactions. And this is the very final slide to leave you with a take-home message. What I've said is that a far from equilibrium environment, the kind of thing we see in these hydrothermal vents, can drive protocell growth. And that protometabolism should be following thermodynamically favored pathways, right up to and including nucleotide synthesis. I've said that positive feedbacks from nucleotides on CO2 fixation, effectively as naked cofactors, should be able to promote faster growth and probably the robust inheritance of nucleotides. But this form of metabolic heredity is not really evolvable. It can gain in complexity by lowering kinetic barriers, but it can't take off in other directions. As soon as we got to ATP, then in principle, we're capable of producing short random polymers, RNA polymers. They would have no information. It would be a random chain of RNA, and we're assuming just a few nucleotides in length. But if we assume biophysical interactions between amino acids and the short RNA sequences, then we have non-random peptides formed through some form of templating. We don't have to specify what that is. We're just assuming that if it's a non-random relationship and the polymerization depends on that surface, then you get a non-random peptide. And their function will depend on their context in the protocell. So if it's a hydrophobic peptide, it would presumably partition to the membrane and potentially promote CO2 fixation. If it's a hydrophilic peptide, then it's potentially going to promote something like RNA replication. And that means that sequences in principle, and we're modeling this as well, should be able to evolve over time to optimize protocell growth from the outset. And that means that information has meaning from the very beginning, meaning in the context of protocell growth. And so we're really trying to get biology and natural selection into the origin of life as early as we can. And from that point of view, protocells becoming cells as soon as they have some kind of genetic code seems to be the most consistent with life as we know it as the way of getting there. Okay, that's it. Pretty much all of this beautiful experimental work has been done by my PhD students and postdocs in the lab. Here are a bunch of them. This is slightly old now, but this is the reverse Krebs cycle that they're holding up. And so I rather like this, but these guys are pretty brave, I would say, doing this kind of work. It's a lot of it doesn't work. I'm only showing you the highlights of the things that has really worked well. It's exciting, but it's also a lot of it doesn't work. Thanks to the funders, and thank you very much for the invitation and for listening. I'm very happy to answer questions.
Thank you very much. And now we have time for questions. Are there any questions?
This is Mark Harbour. I was very interested in your idea about other planets having life if they have these vents. And so what is it about, so you have to have, the ferric compounds, you have to have CO2 or hydrogen.
Yep.
And what about oxygen? Is oxygen a requirement?
More or less the opposite. If there's any oxygen around, it's far more likely to react with hydrogen and prevent formation of organic molecules. So oxygen, I don't know if it needs to be precluded completely, but it's really not helpful having oxygen.
Great. Thank you.
Water, yes, though.
Sure.
Yeah.
Sorry, can it be something else than water on these planets?
It's not something I think about much. In principle, yes, it could. In practice, I don't think we really know why water is so good at what it does. It's able to form all kinds of interesting structures in relation to membranes and so on. As fixed water, it's an extraordinary solvent. I think perhaps the most important thing is it's ubiquitous. It's found almost everywhere. So it's possible to imagine that you could have liquid methane, or you could have liquid ammonia or something like that, but I doubt that it would be as good as water. It would be less common than water. And one of the important points about water is it's driving this serpentinization reaction, so it's producing hydrogen. And again, it's presumably possible to do that from methane or from ammonia, but I don't know that we know of processes that specifically do that. So I would say water is central, but that's more of an assertion than a statement of fact.
So would you say that evolving life would be more possible in any other solar systems, Goldilocks zone, than on our own, since we know for sure that, or almost for sure, that we don't have water in the neighboring planets?
Well, we have water on the moons of Saturn and Jupiter. So Enceladus and Europa. Europa has an ocean, a liquid ocean, which is larger than all the water on Earth. And it's far outside the traditional Goldilocks zone. But because, again, of the properties of water, because ice is less dense than water, you're able to form an icy crust with liquid water underneath it. Whether other things do that, I'm not aware of anything else that does that in the same way. But what that means is, at least for bacteria, so long as you have active hydrothermal systems, and the ones on Europa and Enceladus are produced by the gravitational pull of the giant planets, then we can be way outside the traditional Goldilocks zone and still have the right conditions for life starting. So if I were to go anywhere in our solar system, Enceladus is the place I would be most interested to see. Mars, it seems to me now, it's highly unlikely to have anything on it now. It'll be quite disappointing if 3 or 4 billion years ago, when there were oceans on Mars, that we didn't see the same kind of conditions. So it will be disappointing if life had never evolved on Mars. It will be quite surprising if there's anything left now, but if there is, it's presumably a few meters down and pretty much dormant most of the time. Otherwise, we would see some sign of it in atmospheric disequilibria.
Thank you.
And following up on that, I have a question. So what you describe here quite convincingly, for me at least, was not really knowledgeable in this, but it seems that what you're describing is not really incidental, quite the opposite. It's like a thermodynamically and energetically and inevitable processes that will lead to this protometabolism and protocells. And so basically you can predict that once these conditions exist in other planets, then at least these protocells and proto-life can arise. But the question in Earth, we have two main branches, as you described, the bacteria and the archaea. And so would you have a prediction whether in other planets life, or this prokaryote life, would be similar more to bacteria, to archaea, or maybe in other planets you would have 5 or 10 different branches of these prokaryotes?
A very interesting question. My feeling is that in some sense, bacteria and archaea are very similar to each other. They're similar in their size, they're similar in their morphological complexity, they're similar in their topology and overall cell structure, they're identical in their genetic code and so on. So I would expect probably something similar to that elsewhere, whether you would say they're archaea or bacteria or something else, but I would expect that they would be small, morphologically relatively simple cells. The origin of eukaryotes is a completely separate question and quite an interesting and difficult one. But that happened once on Earth, at least from the phylogenetic evidence that we have. So then the interesting question is, why do we see such differences between bacteria and archaea? And we do have a paper on this from some years ago, where we were thinking about what are the conditions required to take advantage of natural proton gradients in hydrothermal vents? Because the bioenergeticists have been, some of them have been critical of these ideas on the grounds that if you don't have active pumps, then a proton will come in, and if it can't leave again, if it's not actively pumped out, then you'll collapse the driving force. You'll effectively equilibrate between one side of the membrane and the other. Now, that doesn't happen if you don't have a closed cell. If effectively you've just got a single barrier and things come across it and move on. If you've got a closed cell, then it doesn't necessarily happen if that cell is leaky to protons. So if protons come in and then can be carried away or neutralized by hydroxide ions, then we're not going to accumulate protons, and we're not necessarily going to accumulate a charge either, because you have mixing in the system somewhere, which would dissipate the charge. So it all works, and this was theoretical modeling in principle. But then you're left with an interesting different problem, which is, okay, so let's assume that this has taken us through to the level that we have genes, we have proteins, and now we need to pump. If we want to leave a vent, or we want to survive in areas of the vent that we couldn't survive in before, we're going to need to pump. And if you've got a leaky membrane, then pumping across it is a waste of time. Effectively, you pump a proton across it and it comes straight back in. So how would you what would be the selective drivers for the origin of pumping? And the answer that we came up with was, well, you can't do it by pumping protons, but if you have a sodium proton antiporter, it's the properties of membranes in relation to ions are very different. So sodium ions are about 6 orders of magnitude less permeable across fatty acid membranes than protons are. And that means that if you have a sodium proton antiporter, for every proton you pump out, if it comes back in through the antiporter, a sodium ion goes out, then you generate a biochemical sodium gradient, which is added to your geochemical proton gradient. And that gives you effectively, as soon as you have an antiporter, and these are universally conserved across life again, then now you have a suitable selective context for improving your membrane and evolving pumps. And our argument was that this happened effectively simultaneously in bacteria and archaea, but differently, so independently in the bacteria and the archaea. And once they've done that and escaped from event system, then presumably they would take over the world very quickly and preclude any other multiple origins of similar types of prokaryote. This is hand waving. There's no way of knowing if there's any truth in that or not, but it does quite elegantly, I think, answer a question as to why would it happen twice, but not more than twice and not just once.
Okay, thank you.
Hi,
Yeah.
but first, thanks for this fantastic talk. And
Thank you.
I don't mean to be blunt, but I have to say that I read all your books and of course I love them.
Thank you.
Can't say that I understood all of them, but it was a treat. So from a more evolutionary perspective, so it will appear that in most of the time there are really well-definable major transitions in the evolution of complex life. But of course, there is this still one of the most important questions regarding the very first step between the protocellular and cellular forms to arise. With this regard, so how do you imagine this first major transition, and especially in terms of, if you are thinking about a bit more physically terms, in terms of phase-space shifts in energy and information flow?
So I suppose what I've been trying to argue is that protocells come first. And by protocell, I'm thinking quite specifically of fatty acid or mixed amphiphile membranes with things in those membranes that can drive CO2 fixation. So it's an autotrophic protocell, and it's going to be forming the core of biochemistry inside. And what that does is promote growth, because you're forming organic molecules inside and they will follow their biophysical properties. So fatty acids will go to the membrane, amino acids will interact with either the membrane or they will interact with ions in the solution inside, and so on. So I'm assuming that various catalytic feedbacks, which can in principle be inherited, can lead to, I would say, a complexification of that system. It's not really evolvable because there is no real information. All it's ever doing is breaking down kinetic barriers to thermodynamically favored reactions, but it can get better at doing that. So I would say from that point of view that the first major transition then would be the polymerization of RNA inside that context. So now we're adding something which is truly evolvable, which is to say a sequence. And as soon as we can select for that sequence and that is inherited, then we have an evolvable system in a way which it's up to that point, it's just thermodynamics. And that, I mean, I have to say, I find it unsettling because it basically says that thermodynamics is going to give you most of biochemistry right up to nucleotide synthesis, which is hard to believe. It's hard to believe anything else as well. And in the end, I think it boils down to, well, let's do the experiments and see, should we believe it or not? In principle, this can happen. In practice, it's hard to believe. Experimentally, let's see what it says. But if it's true, then the first major transition would be the polymerization of RNA inside a growing protocell, which is then capable of inheriting the RNA sequence and evolving that RNA sequence from generation to generation. So effectively, we have biology as we know it then.
Right. I think it was Brooks who argued that one of the major players here would have been, as you actually stated multiple times, I guess, so the internalization of the energetic processes. So probably this significantly preluded, I mean, not preluded, but it was the first step required, the internalization of energetic processes to the emerging of informational systems that can play practically in symbiosis with these processes.
To me, yes. I think probably the dominant idea in the field still at the moment would be, I mentioned at the beginning, the terrestrial geothermal systems, where in effect, it predicts more of a heterotrophic origin of life. It predicts that various different environments will synthesize various different things like nucleotides or whatever it may be, and that they will come together initially in an RNA world. In other words, information is coming before protocells or anything else, and that it's the information networks which are generating metabolism. And so that's giving you a different order of things. And it comes up with a specific problem about how these how does information accumulate, really? It's quite a difficult problem that field, I would say, has been stuck in for a while. And the nice thing about seeing an autotrophic origin, it has this question mark about can you really do all of this biochemistry without any genes? But if you can, then what it's giving you is basically a lifelike system, which is growing cells, and you introduce into that the first informational molecules, which can be completely random, but so long as they're capable of being inherited, they can change, and we have selection as we know it. So yes, I wouldn't say that this is a particularly new idea, but it's not the dominant idea in the field, and I think it answers the origin of selection in a way that I think most biologists would be more happy with than an RNA world where it raises these questions about information that are quite difficult.
Just a very quick follow-up on that.
Yeah.
So based on the last few slides, you showed in this, so practically biophysical properties of the different molecules really strongly determine the composition and the potential roles that they can play. So this could, in principle, hint at that practically the first platform of information was truly the interaction of molecules with their environment in biophysical.
To my mind, yes, it makes sense, but lots of people have tried to demonstrate these direct biophysical interactions over decades. Some people have claimed that it works, other people have claimed that it doesn't work. They're obviously weak if they exist at all. It may be that they're slightly more complex than just a kind of a naked anticodon and a single amino acid. It may be that you require a small aptamer and that there's some shape requirements, for example. It's the more you get into the details of it, the more difficult it becomes. So experimentally, yes, we are looking at it, and we've not had much success yet either. Plainly, there are not, these relationships are suggested by the code, but they're not easy to measure experimentally.
Thank you very much.
Any other questions? Seems not.
While the others are thinking, I have another question.
Okay.
So in all multicellular life, they share a common signaling pathway that serves like an internal fuel gauge, basically, that is very sensitive to amino acids, basically nutritional conditions. But this system, the mTOR system, is not equally sensitive to all amino acids. There are some particular amino acids that you know, is more sensitive to. And so, based on these biochemical properties, could you predict that what are the important amino acids to pay attention to in a very early life? Or
I don't think I can begin to answer that question. Whether there's any overlap, I mean, I think animals are heterotrophs and eat their amino acids. So I suppose a lot will depend on what is the balance of whatever they're eating. And we get into medical areas of essential or non-essential amino acids. Mm-hmm. For the origin of life, we're thinking about the number of steps from CO2 fixation, and we have effectively about 10 amino acids, which we think, and other people agree from other points of view, are probably the most ancient amino acids. The outlier in there is cysteine, which we have a need for very early on, and is not so easy to make. We're trying to make it as well, but it's not really being done in a persuasive way as prebiotic chemistry. Now, how much overlap there is between these kind of core amino acids and later additions, I don't know. My instinct says that animals are so much later and live in such a different way that there's no requirement for there to be any overlap at all. But if you were to come back to me and say, well, there is an overlap and here it is, then that would be extremely interesting. I'm not aware of an overlap, and I can't think of an obvious kind of biophysical reason why there should be. Mm-hmm. But it would certainly be interesting if there was one. I mean, I think there's been as well interesting selections against amino acids like methionine, for example, which are relatively easily oxidized, and the more aerobic the animals are, the less methionine they tend to have. Also, other systematic, I was involved with some Japanese authors on a paper about the hydrophobicity of respiratory complexes in the mitochondria. Mm-hmm. And the proportion of very hydrophobic amino acids in the respiratory proteins declines with increasing aerobic capacity, and they're replaced with serine and threonine mostly, which are capable of kind of fairly loose hydrogen bonding networks, which presumably facilitate changing conformational states at a faster rate than hydrophobic interactions would, but I have no idea what the reasons are. So again, my sense is that as soon as we have oxygen and as soon as we have selection for higher aerobic capacity and the ideal amino acid repertoires for that, then we would almost certainly overwrite anything that goes back 4 billion years to an utterly different environment where there wasn't any oxygen.
Sure. Okay, thank you.
Other questions? Okay, so I think it's time to conclude. Professor Lane, thank you for accepting our invitation.
Oh, thank you.
It was really cool having you here. As Adam said, you are basically a superstar for our generation.
Quite kind of strange from my point of view, but I'm very flattered.
Yes, thank you very much, and we're really looking forward to reading your new book. Any ideas when? So you said that it
should be out in May, I hope. But it will be my pleasure to send you a copy.
Okay, thank you very much. We're really looking forward to reading that, and thanks again for the invitation, for the audience, for being here, and
thank you.
Nice rest of the day. Take care. Bye bye.
Thanks a lot. Bye now. Thank you.
Bye bye.